Light is truly a fascinating thing. It allows us to see and illuminates the world around us, but its uses go far further than that. The light produced here at Lawrence Berkeley National Labs ALS spans the electromagnetic spectrum from infrared to X-rays, with the goal of utilizing many techniques that harness this light for research in biology, chemistry, physics, and the environmental sciences. For instance, two techniques that are commonly used here are X-ray absorption spectroscopy and X-ray fluorescence spectroscopy. As you can guess from the names of the techniques, they really focus on the X-ray portion of the electromagnetic spectrum. X-rays are useful for penetrating substances' surfaces without harming them, which makes them useful for understanding the inside structure of those substances. This is because X-ray light is invisible and non-destructive, which means they can be used to peer into substances without fear of obstruction or destruction. You could even use X-ray light to look inside of matter performing work, like looking inside a battery that's in use. Let's take a look at the two techniques in closer detail. X-ray absorption spectroscopy specifically uses X-ray light for determining the chemical state of matter. Because this technique requires precise control over a broad spectrum of X-ray light, experiments involving it are usually performed at a synchrotron light source like our very own ALS. One such experiment here that's made use of X-ray absorption spectroscopy is developing horizontal levees to remove nitrogen from wastewater. Long story short, treated municipal wastewater often contains nitrogen, which has been linked to algal blooms that can devastate coastal ecosystems. However, recently researchers have found that horizontal levees can help to remove nitrogen from said water. The specific type of X-ray absorption spectroscopy used in this research is X-ray absorption near edge structure spectroscopy, or XANES spectroscopy for short. XANES spectroscopy specifically focuses on matter that's bonded to the surface of other matter. This type of spectroscopy has been used to understand the distribution of mineral groups, notably sulfides, in the water, biomass, and soil samples taken over 2 years for the levees research. Another research project that's made use of XANES spectroscopy is the survival of T-Rex microvascular structures from deep time. For a long time, the standard has been that protein-based body parts can't survive more than 1 million years, but recently our very own ALS has helped to prove that's not true. XANES spectroscopy aided in discovering the presence of finely crystalline geophyte within a 66 million year old T-Rex tibia, which is a product of protein fusing processes. X-ray fluorescence spectroscopy specifically uses X-rays for determining the elemental composition of materials. In the previously mentioned T-Rex project, X-ray fluorescence spectroscopy was used to map out the distribution and types of metal present in T-Rex vessels, which helps support the researchers' hypothesis about how the T-Rex tissue was preserved. These are just a few things that can be done using the light generated here at Lawrence Berkeley National Labs Advanced Light Source.